Hey guys, today we are going to talk about mutations. So you've already learned a little bit about mutations when we did our evolution unit. So some of this will be a bit of a review, some will be new. Make sure you're taking notes though as we go. Okay, so mutation is simply by definition a change in the DNA, right? So we learned about DNA in our earlier standards, and we know that DNA has a code that will tell the cell what proteins to make, and a mutation is going to change the code. So just like a typo in your paper would change the meaning of your sentence, right? A mutation in the DNA will change the instructions for actually building a protein. And remember, somatic cells, mutations will affect only you. So a somatic cell is a non-sex cell, right? So those would be mutations in your skin cells because you're outside in the sun too much, could give you skin cancer, but it's only going to impact you. In sex cells or gametes, it's going to affect your offspring, right? And these were the kind of mutations that mattered for evolution because they're passed on. And since they're being passed on to the next generation, they can lead to genetic disorders. And remember, mutations can be positive or negative. So in this unit, we're going to focus a lot on the negative side of mutations because you're all talking about a genetic disorder, which results from a mutation. And genetic disorders are negative, right? They're not good to have. But keep in mind they can be positive, right? So think back to our evolution unit. And remember, a mutation can also make you more fit for your environment. So keep in mind they can be both, right? Even though our focus will be on negative. So mutations will change the genotype of an individual. And remember, genotype means their genetic makeup, right? So a mutation in the DNA changes your genetic makeup, right? It's going to make your DNA read something different. Those mutations can possibly change your phenotype, which is your physical makeup. Remember, sometimes phenotype are traits you can see, like hair color, eye color. It could be a short stature or tall stature. It could be long fingers, a long face, right? But it can also change things you can't necessarily see, but how your body functions, right? So it can change the structure of your heart so it doesn't beat right. Or it can change the muscles in your back so that your spine starts to curve. So those are also phenotypes. So there are two main types of mutations. There are gene mutations and there are chromosomal mutations. So we're going to talk about both. So a chromosomal mutation is a mutation that affects an entire chromosome. So remember, you have 23 pairs of chromosomes, 46 total, right? And on those chromosomes, you have thousands of genes. So if you have a chromosomal mutation, because it affects an entire chromosome, it can affect thousands of genes. So most chromosomal mutations are super dangerous and very few are possible to even live with. In the picture, you can see some examples of chromosomal mutations. Deletion is where a big chunk of your chromosome is removed. Duplication is when you have a double part of the chromosome, so you have extra genes that you shouldn't have. Inversion is when parts of your DNA switches, right? So your chromosome switches around and you could cut off a part of a gene and hook it to the wrong gene. And then translocation is when two chromosomes will actually cross and exchange parts of themselves on accident. So these are all really bad and really dangerous. Gene mutations, on the other hand, affect only one gene. So for most of your genetic disorders, you're going to have a gene mutation. It's going to affect just one gene. And these are more common mutations because you can live with them, right? So lots of people have gene mutations that cause small issues or one genetic disorder, right? But it's possible to live with them. So gene mutations are common. And you can see here there are two main types of gene mutations: point mutations and frameshift mutations. So there are three types of point mutations. The first is a silent mutation. And you can see in the picture here, a silent mutation would switch this C to a T, which would switch the RNA from an AAG to an AAA. But if you use that wheel during translation, right, you would get the same amino acid, lysosine, right? So this is a silent mutation. The DNA is different, but because the protein is built the same, it's not going to matter, right? That's a silent mutation. The next is an early stop, right? So if the original DNA was TTC, the early stop point mutation may change it to an ATC, which when transcribed becomes UAG, which is a stop codon, right? So in the normal protein, you might have thousands of more amino acids that's supposed to come after that point, but because of the mutation, now you're going to have this early stop, right? Your protein's going to be way too short. It's not going to have everything it needs, so it's not going to function right. And then the last type of point mutation is called a substitution or a missense mutation. So this is where you have one amino acid changed to another, right? So we had a TTC, now we have TCC, and that's going to change this amino acid, right? And you can see there's kind of bigger or smaller mutations. This amino acid looks pretty similar to the original, so it's not as bad as, say, this mutation. But again, a substitution would just be swapping out one little nucleotide and getting a whole new amino acid. So all point mutations involve swapping just one little nucleotide, right? You're swapping an A for a C, something like that. So it's just a one switch in that DNA. So point mutations happen quite a bit. And again, 3 types of point mutations. The other kind of gene mutations are called frameshift mutations. These mutations are going to add in an extra base or take out one of the bases. And they're called frameshift because they're going to shift the entire reading frame. All right, so look at this first picture here, right? The normal reading frame is this, right? We read in 3s, so CAG, CCC, ACT, right, to make this amino acid sequence. This is this is an insertion, right? We've inserted an extra base here, T. And what that does is it shifts that reading frame, right? So this CCC is messed up because now it's TCC, but this ACT is also messed up, right? Because now you get this C from here comes down with the AC, and that T goes to the next one. So it's going to bump every single amino acid after that mutation. So it's going to be messed up all the way down the chain, right? Which is different than our substitutions because they only affected it at one spot, right? Versus these frameshift affect every single amino acid after that mutation. And you can see in the lower picture there, you have a deletion. So right here, you should have had an AAA, right? One of those A's got deleted on accident. So now you have an AAC, right? And again, you affect every single amino acid after it, not just one is mutated now, the whole rest of the amino acid chain is. So frameshift mutations are more severe than just a point mutation, right? But both of those are types of gene mutations, which are less severe than chromosomal mutations. Okay, so feel free to watch this video again if you're still not sure about it or ask in class. We will go over this tomorrow, so we'll have a little bit more information coming at you. Thanks, guys.